Okay, there are a couple of important things that are happening during this electron transport chain. Okay, so there is a reason why it is named electron transport chain, because electrons are being shuffled through this chain of proteins. More details in the next couple of slides, but electrons are being shuffled through this chain of transmembrane proteins. And as they're being shuffled through, every time a photosystem or cytochrome complex or photosystem 1 gets those two electrons, it is shuffling two hydrogens from the stroma into the lumen side. So these transmembrane complexes are really multitasking in the sense that as soon as they get those two electrons, they are pumping two hydrogens from the stroma into the lumen side. So they are pumping protons against their concentration gradient. That is why they need energy to do that. And what you can see in this photosynthetic electron transport chain is that plants use the energy of light to first and foremost split water right here, but they also use the energy of light to kind of raise those electrons into their high energy state, and then they can flow simultaneously to the next complex. And then they use energy again to raise those electrons to their excited state, and then they will fall down simultaneously where the ferredoxin is. Photosystem 2 absorbs light from 680 nanometers, and photosystem 1 absorbs light from 700 nanometers. So what is inside the photosystem? These chlorophyll molecules are in the center, and that's why this is called a reaction center. Always the donor of electrons will be water. Okay, and then you have here around the reaction center, what you can see are those antenna chlorophylls. So all those carotenoids and luteins, they're kind of helping guide the light toward the reaction center. And always in the reaction center, what you can see in the photosystem 1 of the reaction center, what we mentioned is that they absorb light that is of 700 nanometers, and in the photosystem 2, the reaction center is absorbing light from 780 nanometers. So right here in the middle of the reaction center, you actually have two molecules of chlorophyll called chlorophyll dimer. Around that, you have the antenna chlorophyll, which kind of guide the light towards the reaction center, where water is going to be split, and then the electrons will go through the reaction center, and then they will find their way to the next protein in the electron transport chain. Okay, so let's talk about what is the pathway of these electrons. So from water, the electrons will be centered in the reaction center of the photosystem 2. Then they will be given to this molecule called pheophytin. Then on the molecule called quinone A, quinone B. Then they will be carried through the membrane with a molecule that is called plastoquinone. Plastoquinone. Then through the iron core of the cytochrome complex. Then they will go on to another transmembrane protein, which is called plastocyanine, and then they will be transferred to the reaction center of the photosystem 1, which will then carry them to the ferredoxin, and then finally they will be attached to the NADPH. So there is the picture one more time. So what we have talked about so far is what was going on the surface of the thylakoid membrane, which was a photosynthetic electron transport chain. So right here, you see we've created the ATP and we've created the NADPH, and so now we can move on talking about Calvin cycle. Calvin cycle was discovered by a guy called Melvin Calvin. So there he is, right there. So he was a chemist. So he worked a lot, and he pretty much also discovered all the stages of the Calvin cycle. Sometimes it is called photosynthetic carbon reduction cycle. But I think we should call it Calvin cycle because just, to honor the guy who discovered that cycle. So what you can see here is there are three stages. Carboxylation is where carbon dioxide is being absorbed, reduction stage, and then regeneration stage. So in the first stage, we need to attach this carbon onto something. And as I mentioned before, this carbon is being attached to ribulose 1,5-biphosphate, which is then converted into two molecules of a 3-PG. In the process of the reduction, this is where we are going to use those energy that we created in the light reactions, ATP and NADPH, so that we can create those molecules that have 3 carbons, so those trio sugars. And then in the process of regenerations, we need to, when I say we, I mean me and plants. So in the process of regeneration, what needs to happen is we need to have those supplies of RUBP, because if we don't have enough supplies of ribulose 1,5-biphosphate, then there will be nothing that the CO2 can be attached to. So this process of regeneration is extremely important, and you will see how plants always make sure to have enough RUBP. Now, this is kind of a simplified cycle where you can see ribulose 1,5-biphosphate, and then carbon dioxide is being attached to that, and then you have the 3 carbon molecules, the 3 phosphoglycerides, so 3-PGs, which are then converted into 2 different types of molecules, because only this molecule, 1,3-biphosphoglycerate, will actually be exported out. The other type, glyceraldehyde 3-phosphate, or G3P, will be used to regenerate ribulose 1,5-biphosphate. Now, if you are if you like have a very good grade in chemistry and you're a chemistry bug, you will see that there is something wrong with this picture. So the names are correct, and the structures of the molecules are correct. But like I said, if you kind of turn on your brain a little bit, you are gonna start to count carbon molecules, and then you will see that there is something seriously wrong with this picture, because these black dots, so these are carbons. So if you count them here, 1, 2, 3, 4, 5, and then you add 1, okay, so now we have 6. 1, 2, 3, 1, 2, 3. Okay, so we are good here. So how do we go from a 6 carbons in 2 different molecules back to 5 carbon molecule? Okay, so you will be thinking like there is something wrong with this, and yes, there is something seriously wrong with this picture, because it is not 1 molecule of carbon dioxide that's being absorbed. It is 3 molecules of carbon dioxide which are being added on. And you will actually have 6 molecules of G3P, and only 1 of those will be exported out to create sugar. All of the rest of the G3P, so the 5 of those, will be used to regenerate ribulose 1,5-biphosphate. So we see like all of those sugars are being created, but only one will be exported out. So you can see how it is very important for plants to have lots of supplies of RUBP. So, and now when you actually count, I'm not going to do this in this presentation because, I'm trying to keep the presentation short so they can be uploaded to YouTube. But when you actually do count all of the carbon molecules, you will see that now this cycle, makes sense. So please remember that it is actually 3 molecules of CO2 which are being absorbed. And the enzyme that is responsible for fixating carbon, it is called Rubisco. It stands for ribulose 1,5-biphosphate carboxylase oxygenase. So it means that it can absorb both carbon dioxide and oxygen. We will talk about this in plant physiology in details, but it's a very complex, most abundant protein on Earth. So plant has a lot of them, but it's a little bit faulty. It can accidentally absorb oxygen, but I'm not going to go into details. For your level, this is just enough. It just so you know that it's called Rubisco, and it has a copper core. And it is responsible for fixating the carbon. Okay, so let's talk about next what is going to happen. Please make sure that you have access to Moodle. Our next quiz, which will be multiple choice questions, will be on April the 5th. Only 2 units, leaf and photosynthesis. So what I've lectured about leaves and what I lectured about photosynthesis, I think you all have the book as well. The quiz will be completely on Moodle, and it will be timed to 1.5 hours. So I think you have plenty, time to finish those multiple choice questions. You already know how they look like from quiz 1. So, the type of questions will be very similar, okay? Please make sure that you log on. The only thing I don't have right now is I don't have the actual date. I'm sorry, I don't have the actual time when the quiz will start. But I will update you on that in the announcements. Okay, so please now just remember this. Okay, so there will be a quiz on April the 5th. On leaf and photosynthesis. If you guys have any questions so far, please feel free to email me or let me know if I have to go back and explain something in more detail. Okay? Have a good one, guys.